Hello, and thank you for joining us. Today, DILIsym's Chief Scientific Officer, Scott Q Siler, will describe the features and use of IPFsym version 1A to support the development of effective treatments for IPF patients. While a few of you are still joining in, let's review a couple housekeeping notes. We take your privacy rights seriously, and by attending this event or participating in the Q&A session, you are allowing us to contact you for follow-up. You may ask questions via the questions panel on your dashboard at any time, and we encourage you to do so. A Q&A session with speakers will follow the last presentation. Now, a quick question for everyone in the audience before we begin. Does your organization have an internal QSP modeling group? Please select one of the choices below. We'll give you just a few more seconds here. Great. So it looks like 67% of you do have an internal QSP modeling group. Excellent. I'm excited to have you learn more. Now, a little bit about my friend and today's presenter, Scott Q Siler. Along with developing and using DILIsym software to improve the development of safer drug therapies, he's also an associate director of the DILIsym initiative since its inception in 2011. Boy, that seems like a long time ago. He helps oversee the development of DILIsym software suite and is also a project manager for DILIsym service projects contracted by pharmaceutical companies. Dr. Siler graduated with a PhD in nutrition from the University of California, Berkeley, and a Bachelor of Arts in biochemistry and molecular biology from the University of California, Santa Cruz. Please help me welcome to the virtual table, Scott Q Siler. Scott, take it away.
Thank you, Arlene, and go slugs. We'll first start off just a brief disclaimer. Effectively, to summarize, I am not dispensing medical advice here. And then for our agenda, we'll give you just a brief introduction to our division of Simulations Plus, the DILIsym Services Division. And then we'll move into, I think, what most of you are here for, to learn a bit more about IPFsym and our first version of this platform, this QSP model. And then taking it a step further, how we simulate treatment, including the standards of care, Nintedanib, Pirfenidone for IPFsym. And then we'll conclude with just kind of some information about licensing and services of this model, and then open the floor for questions and answers, the extent of which obviously is time permitting. Arlene, I think you have oh no, we already did the poll, sorry. I got a little behind there. Okay, so here on slide 4, one of the things that I think we're really excited about, our division, DILIsym Services, is being a part of the Simulations Plus family. I think it's about 3.5 years now that we joined Simulations Plus. And one of the things that we saw and motivated us to participate in this broader organization is kind of the way the puzzle pieces fit together. Multiple different modeling approaches that were really complementary and enable each other discipline to kind of rise even higher. So we really feel like collectively we do a fantastic simulation-based science, as well as kind of supporting business objectives, both obviously our own, but those with whom we collaborate, those with whom we partner. And it's also worth pointing out that really it's kind of the solutions, the science is applicable all throughout the pipeline. Depending on what the nature of the question is, we have services and products that can certainly support all of the above. For our specific division, really focused on QSP modeling, We have several products that are either already available or in development, and you can see them listed here. Obviously, the one that kind of our feature, the one that launched the enterprise, so to speak, is the drug-induced liver injury simulation platform, or DILIsym. And from there, we've kind of built kind of on the core capabilities, core competencies, and really extended our reach into a variety of other areas. Collectively, as part of Simulations Plus, we all really believe in the mission statement that's listed here, which, kind of cutting to the chase, we believe in innovative solutions, diversity and inclusiveness, accurate, effective communication, and quality products and services across the board, not just our division, but all of Simulations Plus. And then coming back to the kind of the QSP offerings, I mentioned, of course, DILIsym, and certainly that software is available for licensing, as is NAFLDsym. So that's a QSP model of non-alcoholic fatty liver disease and NASH, non-alcoholic steatohepatitis. We're going to discuss today the platform from idiopathic pulmonary fibrosis, and then two other models that are in development and probably in the queue to be released in the relatively near future are RENAsym, so that's acute toxicity of the kidney due to drugs, and RADAsym, acute radiation exposure and the consequences of that. Another really interesting development in terms of what we're seeing from the marketplace, from pharmaceutical industry, is really frequent requests for us to consider developing new QSP models and to really support specific programs by developing these custom QSP models. And what I've found over the years is that the more tools as modelers, as QSP modelers we have in our tool belt, the more able we are to really support pharmaceutical development across a variety of different platforms. And so really that's, I think, a nice little development in that regard. So our division, the DILIsym Services Division, relatively small, about 20 or so of us, and really interdisciplinary. From myself, as Arlene mentioned, I'm originally trained as a nutritional biochemist. Of course, I've been doing QSP modeling for some 21, 22 years now, something like that. And so I'm kind of somewhere in the middle. But we also have, on our team, immunologists, mathematicians, physicists, chemical engineers, pharmacologists, and the list goes on. So really, we kind of come together to really allow us to, analyze the science, put it together with the math, and make predictions that really potentially impact programs. The other thing I'd like to point out here is there are a number of individuals on this slide who have contributed to the development of IPFsym. And so I want to make sure that, whilst I'm the one presenting it, really it's been very much a group effort, a very collection of individuals that were really necessary. Right, that interdisciplinary interaction was required in able to do that. So another thing that I found, and I'm actually really excited to learn that most of our audience does have QSP groups. But one thing I found in this, being kind of one of the earlier practitioners of QSP modeling, is oftentimes everybody has their own kind of unique definition of it, their unique understanding of it. So just to make sure that throughout the presentation, you understand what our definition is, our lexicon, I figured it was worthwhile to take a couple of minutes to just describe it. So really, for all QSP models, the foundation is kind of the mechanistic mathematical representation of the underlying biochemistry. Obviously, in the disease state, that's really focused on the pathophysiology. What are the relevant pathophysiologic relevant biochemical components of the disease state? And those need to be captured mathematically in order to make sure then that you are able to represent the disease state. And then, once you have that disease adequately captured from that mechanistic mathematical modeling point of view, obviously the clear next step is to try to evaluate modes of treatment, modalities of treatment. And so then we need to superimpose upon that predictions of exposure of a compound. And in this area in particular, oftentimes the targets are intracellular. And so really, if you want to be able to predict the compound exposure at the site of the target, then really you're talking about employing PBPK modeling. And so again, you can see how within Simulations Plus, we're now talking about putting puzzle pieces together as we kind of now align Gastro Plus PBPK predictions with IPFsym QSP predictions. And then superimposed upon that are obviously the ability for the compound to interact with the drug targets. And actually, it goes a step beyond that. So beyond kind of strict PD, where the interaction of the drug with the target, but really quantifying then the mechanisms of action. In other words, how does the drug and its target interact with the underlying pathways, the key pathways of the disease? Those are the mechanisms of action, really. And so a real good QSP model is flexible enough to allow kind of novel targets, novel mechanisms of action to be represented. And so across the board, as you kind of put together this Venn diagram, then obviously it's in the center of that therein lies the ability to predict efficacy. Okay, so let's move into an introduction of IPFsym. But before we do, I believe, Arlene, we have another poll question.
We do. Does your organization have assets being developed to treat IPF patients? This will be an interesting follow-up, Scott. We'll give everyone just a few more seconds here. All right, let's close the polls. Oh, and it's a bit of a split. So we've got 38% with yes answers on organizations having assets being developed to treat IPF patients. Back to you, Scott.
All right, thank you for those poll results. It's also, and it actually reminds me to mention, whilst the focus of our model currently is on idiopathic pulmonary fibrosis, I think what we've come to realize is it's also a nice starting place for other types of pulmonary fibrosis. One example would be what's observed following COVID. And for a number of patients, pulmonary fibrosis has been observed. And so there's the possibility then that maybe we could, starting with IPFsym, build it out and build in some of the unique pathophysiologic aspects of COVID and predict the fibrosis associated with that, and hopefully potential treatments for it as well. Okay, so switching back the focus to IPFsym. So idiopathic pulmonary fibrosis really is a disease of the aged, which is really, I think, an important aspect, an important consideration from the clinical point of view. So a couple of things really manifest there is, one is, I think because of the age of the population being the target, some of the pathophysiology is driven by cellular senescence, and in other words, kind of an inadequate autophagy that leads to cells kind of persisting longer than they should, hence having higher apoptotic rates than they should. And it seems that probably is a primary contributor to kind of keeping the fire going, if you will, from a pathophysiologic point of view. Another interesting aspect is that it takes, I think, a while for the disease to manifest far enough, extensively enough, to really impact the physiology and in particular respiratory function. So upon diagnosis, most patients, it seems, have already progressed pretty substantially, and hence the mortality rates are relatively low. We're talking about an aged population that already has a fairly extensive presence of the disease, and so really, you can see that within 5 years, a vast majority of patients do not live much longer than that of IPF patients. So really, clearly we have a need for treatment to preserve life, to extend quality of life, and all of the above. One of the reasons for the compromised physiologic function, respiratory function, is a change, a disruption, and actually kind of the physical anatomy. And so the fibrosis can be so extensive that upon visual inspection, the lungs look to have this honeycombing sort of presentation. And when a lung is in this honeycomb, a section of the lung is in the honeycomb state, it is acellular and unable to contribute to respiratory function. So again, it's kind of the extreme, if you will, of fibrosis in a portion of the lung. It's a progressive disease, IPF is, and so you can see that the respiratory function declines over time. We'll talk about other aspects of that subsequently. And so really what we've got now is, a pretty high number of patients that, need help. Treatment is certainly warranted, and a pretty substantial effort within the industry to develop antifibrotics. And so a lot of questions about, hey, is this going to work in IPF? Will this work for this aged population, et cetera, et cetera? In other words, a good sort of recipe, if you will, for us to bake a QSP cake in what we're calling IPFsym. And so it has the ability to predict the effects of treatments on fibrosis, inflammation, epithelial cells, all of course within the lungs. We account for interpatient variability with SimPops, and we're poised to release it here in the very near future. Whether it's late Q1 or early Q2 is to be determined as we proceed and kind of finalize our quality control, et cetera. And then, of course, it can be used to support drug development, like most QSP models, really to predict efficacy of a novel target, to really help optimize clinical trials, to identify responsive versus non-responsive patients, treatment paradigms, dosing paradigms, and perhaps most importantly in this area, combinations of treatment, because there are two standards of care already present, albeit with limited efficacy. So staying with the disease, there's a really, I think, fundamental aspect of the disease that really contributed to the design of IPFsym, and it's these three distinct functional zones. So we've already talked about honeycombing a little bit, and so honeycombing is sometimes described as the barren wasteland. It's again acellular. It's nothing but the extracellular matrix, the fibrotic component, and unable to contribute to respiratory function. On the other side of the spectrum is the normal parenchyma. So these are unaffected alveoli, happily contributing to the respiratory function, and really no influence as of yet from kind of the disease state. And then there's that intermediate state where really the action is really happening, and that's the fibroblastic foci. So it's in this specific section of the lungs where you see inflammation is ramped up a bit. You see that the fibroblasts have become activated in synthesizing the extracellular matrix. You also see then beginning to see disruption in the respiratory function as well. And these zones aren't static, as you might guess, since it's the progressive disease where the normal parenchyma transitions to the fibroblastic foci, which transitions to the honeycombed tissue. And so it's also important to point out that there are imaging modalities that allow us to quantify, to see and quantify the proportion of each of these zones in the lungs of IPF patients. So putting all that into practice then within IPFsym, we have our normal parenchyma really focused on those epithelial cells, the AEC type 1 and type 2 cells. And their ability then, by and large, to contribute to normal respiration. But and then we have the fibroblastic foci, where basically it can extend out in the progression paradigm to kind of envelop a portion of the normal parenchyma AEC cells and kind of extend out the reach of this fibroblastic foci. Within the fibroblastic foci, you can see in this diagram, color-coded to some extent, the epithelial cells in gray, the fibroblasts and extracellular matrix, the real fibrotic components, colored in green, and then the inflammatory cells and mediators colored in blue. And it's this tremendous kind of spaghetti of a diagram because of all the interactivity between the types of cells and the mediators and the influence all coming together again to capture the apoptosis of the AEC cells, of the activation of the fibroblasts, the ongoing actual fibrosis synthesis, as well as the inflammation, all of the above. As this progresses and the extent of extracellular matrix and fibrosis accumulates, then those fibroblastic foci transition into the honeycomb tissue. Again, no contribution to respiratory function, as is the case in the actual patients. And then kind of putting all this together, we basically sum up, the contributions from the honeycomb, the fibroblastic foci, and the normal parenchyma for any given simulated patient for any point in time, in order to predict the changes to the forced vital capacity, to the HRCT imaging of the various regions of the DLCO, the 6-minute walk distance. Classic indicators, classic markers of respiratory function and IPF extent, pulmonary fibrosis. So all of that is captured within IPFsym version 1A. Also, it's worth pointing out that as we consider kind of treatments for IPFsym, really by and large, they focus on this fibroblastic foci where all the action is happening. One obvious target is kind of the fibroblasts and preventing them or reducing the number of activated fibroblasts. There are a number of other certain targets within here, but that being one obvious example. And so obviously we have the ability to capture a lot of those. I should also point out that obviously we don't have the full complement of targets out there, but we do have approaches for building in, as I mentioned at the outset, some of the novel targets, the novel pathways, et cetera. And at the end of the presentation, I'll describe at least briefly how we would approach that. Another really important consideration with IPF, as it is with many diseases, is interpatient variability. And so if let's turn our eyes to this lower right-hand corner, this data from Biondini et al. That shows then the decline, the progression over 1 year's period of time in the forced vital capacity for some patients with idiopathic fibrosis that are considered fast progressors and some that are slow progressors. That's defined by kind of a 10% change in FVC over that 1 year period of time. And so you can see then, just looking at the shape of each of these curves, the slope of each of these lines, that they are, there is quite a bit of variability, interpatient variability here. And underlying that respiratory variability are as well a substantial amount of variability in the progression rates from the honeycombed, from the, as well as the fibroblastic foci based on the imaging modalities. And so we obviously then have to, make sure that within the framework that I just described, we have the ability to also recapitulate this sort of variability. And so let me now transition to the next slide to describe how we do that. We use our simulated population or SimPops approach, really deploying methodology that was developed, 20 years ago and has been put in practice consistently. And we're basically employing those same sorts of practices where we're going into the pathophysiology, understanding what are some of the important areas, the important contributors, and then within those areas, what do the available data indicate are the ranges of those pathophysiologic components? What are the ranges of the parameters describing that? And then doing combinatorics for each one of those to generate a unique simulated patient that maybe, in one case has high AEC apoptosis, but low ECM synthesis and degradation. And continue with it, so you have hundreds and hundreds and thousands of patients with all those combinatorics, the highs, the lows, the mids, et cetera. Once we've generated a group of potential simulated patients, the obvious next step is to do a form of validation to really then compare them to a multitude of clinical data to ensure that everybody, all the patients are within the constraints as documented, all kind of at the same time. The final step is to treat all of the simulated patients with the standards of care. So in this case, that's Nintedanib and Pirfenidone, and to make sure that the response is appropriate, that the patients with drug on board are presenting as described in the literature. So we've applied that approach to generate a large number of SimPops patients in kind of what I would describe as various categories. So the first is really just from a modeling point of view, really modeling them at steady state. Even though it's a progressive disease, looking at those parametric combinations and making sure that we are able to generate patients that meet all those constraints I just described allows us to have a sense of, in terms of what those, the various contributions of each of the pathophysiologic components. And then using that as a starting place, we then, we have all the various different patients, and then we employ, activate, if you will, the disease progression paradigm. And so we let them get worse over time, and with, starting with a different set of parameter combinations initially. So we really have kind of a two different axes that we're paying attention to, and hence kind of the ability to pull patients. This is obviously just in the untreated state, from early progression, from late progression, from this set of parameters versus this set of parameters. So it really gives a wide array of options for us to understand and explore with simulations. We do this not only in this untreated state, but also similarly with pirfenidone on board, with nintedanib on board. And so our SimPops include the full panoply of disease progression, as well as kind of parametric combinations representing the differences, the interpatient variability in the pathophysiology. We've done an extensive amount of kind of calibration validation. I will, time only permits us to do a bit of that. And so I'll kind of touch on the ones that are highlighted here in blue. And I should also point out that kind of the validation of the patients, we do all this simultaneously, right? A patient isn't considered valid unless it agrees with all of these data simultaneously. Before we dive into those comparisons, though, I think Arlene wants to have one more poll question.
I do, thank you. All right, everyone, how do you currently estimate the influence of interpatient variability when predicting efficacy for IPF assets? Again, we'll give you a few seconds here to answer. Okay, we're going to close the poll here. And it looks like literature review takes the lead. Back to you, Scott.
All right. Okay, so now let's let's dive into how we've validated our SimPops. First, we'll focus on kind of the immune, the inflammation section, and so we've kind of pulled out the relevant components. Really, that's the neutrophils, the macrophages, and a wide variety of mediators, not only coming from those neutrophils and macrophages, but also being secreted by the fibroblasts and the activated type 2 alveolar epithelial cells. And so we have some nice lung biopsy data that indicate the number of macrophages and neutrophils in the alveoli, and also categorized from kind of the slow progressing versus the fast progressing patients. And so we've taken our SimPops and made sure, again, compared them to these clinical data, and across the board, both for the neutrophils and the macrophages, we are within the clinical data, the range of the clinical data. A couple of additional considerations. First, you'll note that we, in the SimPops data, or results rather, we have two categories. We also have early progression versus late progression. Hopefully my description a couple of slides ago allows you to understand kind of the difference between the two. But also, it's worth noting that, whilst we do I think, a fine job of capturing the range of variability for the neutrophils, for the macrophages, there's obviously still some room for improvement. And this is, one of the things is we have a really nice, efficient process for developing a new IPF SimPops patients. And so we're, kind of on an ongoing rolling basis, trying to expand, obviously the variability here and some of the other areas that we'll be looking at along the way. So again, kind of the takeaway here, I would say, is consistent with, i.e. within the range of the clinical data, but some room for improvement for the macrophages, certainly. Then transitioning from those cells to the actual mediators. And so here you can see there's the mean and kind of the maximum or minimum for each of the mediators, and left to right, there's TNF alpha, TGF beta, MMP, on the top, VEGF, PDGF, and IL-1 beta on the bottom. Also then comparing kind of the early versus the late progression against the mediator data, which were just obviously collected from IPF patients, in this case, not discerning between earlier or late progressor, fast or slow progressors. And as you, again, hopefully your eye is telling you that it's certainly, again, the case that the simulation results from the SimPops are in line with, consistent within the range of the reported clinical data. Again, something like an MMP, it's pretty obvious, maybe not capturing the entirety of the range, but certainly well within the range. And you can see then that's probably the case across the board. As we transition then from the inflammation to the fibrosis area, so again listed here in green, this is really what are the processes that are driving the activation and the proliferation of fibroblasts, including this epithelial mesenchymal transition, or the EMT, and emanating from the AEC2 cells and kind of a phenotypic transition to fibroblasts. And then as well as the synthesis of the components of the ECM, not only the collagens, but elastin and proteoglycans. And in addition to synthesis, obviously the breakdown as well. So the turnover across the board. And so again, thanks to some nice lung biopsy data, we're able to kind of do the same, apply the same approach for total collagen and the proteoglycans to then look at the range of SimPops patients as compared with the clinical data. In this case, and this is just a consequence of the way the clinical data are plotted. So this is mean and standard deviation, just one standard deviation. We've intentionally allowed our SimPops to extend beyond one standard deviation. I think it's 2, maybe 2.5, because in most populations, it certainly while the frequency of the distribution gets less, it still does extend out to those edges. And so we've allowed that certainly to happen in our comparison here too. So again, early to late, all seem to be nicely consistent, not only with the collagen, but also the proteoglycan data from IPF patients. Now, as we transition to some other clinical markers, I mentioned this high resolution computer-aided tomography, the HRCT. So it's a way to kind of image the honeycombed area, the fibroblastic foci, also known as reticular opacity, amongst other aspects of the lungs. And groups like Park et al. Have done a nice job not only of describing the imaging, but quantifying the imaging. And there are a variety of different approaches being applied here, but effectively you can, using a series of scans and then quantifying how much of it is reticular opacity, honeycombing, normal parenchyma, you can then quantify those functional zones in IPF patients. And so we've done this, quantified our predictions of the fraction of lung volume from fibroblastic foci or from honeycombing, and compared it to the data not only from this Park paper, but also this more recent paper by Balestro et al. And again, you can see that, I think two things come to light here. One is the SimPops are within the range of the clinical data, but also you can see that there's a change in kind of the distribution with progression. It looks like, the number of patients with higher fibroblastic foci seems to increase visually. Same thing with the number of patients with a higher degree of honeycombing seems to increase. Again, consistent with the way we understand disease progression. So those are those functional zones. And then as we transition into kind of the respiratory outputs, so forced vital capacity, the DLCO on the bottom. And in this case, these are the percent predicted. So for a given individual of a specific age, gender, and size, what would you expect the FVC to be if absent disease, and then with the influence of IPF, how does it compare? And so you can see then, kind of generally speaking, it's for on average, roughly 60 to 70% is FVC percent predicted in IPF patients, although again, quite a bit of range. And our SimPops certainly consistent with that, as well as pointing out that, again, I think it's obvious to the eye that the influence of progression causes a decline in respiration over time. And, building on what we just saw in the previous slide, as the fibroblastic foci and the honeycombed areas increase, the respiratory function also declines. Same thing, DLCO, which is kind of the ability of the lungs to allow for the diffusion of, in this case, specifically carbon monoxide. That's the test, but it gives you an index of it, obviously O2 and CO2 diffusion as well. And then, so now coming back to those IPF progression rates. So I think I showed these data from Biondini earlier, looking at the fast progressors. Again, that's where the delta FVC is greater than 10%. The slow progressors where it's less than 10%. And you can see by pulling out a subset of our SimPops patients that we really have slopes and that are really nicely consistent with the categories described by Biondini. Again, hopefully I'm showing you all these different axes. We're establishing that we have paid great attention to validating the SimPops across not only the pathophysiology, but also the respiratory and the clinical markers. So at this point, I'd like to just give a very brief introduction into the IPF software. And so it is, as with some of our other offerings, MATLAB based, and we've built a graphical user interface over the top of that. I should point out that certainly a user has the ability to, if you're a MATLAB wizard, to run simulations via the command line or to use the graphical interface. The GUI is nice in that it allows us, as you can see, to kind of gather and visually see specific parameter sets and which of the value sets you're using. In this case, you can see this is one of those simulated patients, a representative patient 2, no treatment, simulated for a full year, and that took 3 to 4 minutes on this computer. Depending on the computer, it could be faster or slower. And then we have a variety of ways to, once we have the simulation results in the workspace, to visualize them. Obviously, you can use a wide range of tools, including exporting to Excel within the program itself, which is as you're doing the modeling kind of real time, a nice convenient way to do it. You can look at some specific outputs. Since we were talking about forced vital capacity, from the pull down list, we can select that and plot that, and you can see that there's a reduction, albeit a slight one, over time in this patient. Let's reset and looked at the honeycombed tissue, that dynamic fraction, and you can see as well that it's increasing, albeit at a relatively slow rate over the course of that year. Many different aspects of simulations, of course, types of questions that we could ask here. One thing I'm going to set up to run as we return back to the slides, shouldn't take very long, is, let's see, we'll do run in parallel. This same patient, representative patient number 2, with Nintedanib, with Pirfenidone, and untreated. And so we'll be able to compare all 3 of those. We don't need a time report, and in this case, we won't save the results because we don't need them going forward, but we will save all the outputs, and we'll start that simulation. And as we do that, now we'll return back to the slides, and we'll come back to that in just a few minutes. So giving you an overview of our SimPops patients and kind of the underlying pathophysiology in the untreated state. So now simulating treatment in IPF starts with the standards of care. And so Pirfenidone is one of those, made by Roche Genentech, a small molecule, been on the market for a few years now, and really has, the phase 3 data show, does a nice job of slowing down disease progression. Neither Pirfenidone nor Nintedanib completely arrest progression, but it does slow it down. So it's certainly helpful, but also room for additional contributions from further treatment approaches. Looking at the kind of the mechanisms of action, Pirfenidone, actually, this is applicable to both Pirfenidone and Nintedanib. There are a lot of data indicating it could do this, it could do that. And if you even talk to the folks who are real experts in each of the compounds, you'll see in reviews, we're not really sure what the mechanism of action is. So kind of distilling what's in the literature, those specific mechanisms that really seem to be have clear effects. A, the data are really irrefutable, and B, impactful mechanisms are the inhibition of fibroblast activation for pirfenidone, in addition to directly inhibiting the synthesis of the procollagen. So that's on top of the changes to the fibroblasts. And so we've implemented each of those effects consistent with the way the data were described by the two citations here. Then we simulated effectively this phase 3 trial. And so you can see the simulation results compared to those clinical data in this figure here on 29, where initially what we did is we selected out a subset of patients, sim cohorts that aligned with this much more rapid rate of disease progression in the untreated state, the placebo group. And then we applied those mechanisms of action of pirfenidone to restrict those specific pathways and predicted the change in FVC. And you can see a real nice alignment between the clinical data and the simulation results for our simulations of pirfenidone. We applied this same sort of approach with, before I get to the Nintedanib, I wanted to just also show, interestingly, this Biondini paper also describes not only the disease progression in the absence of treatment, but also with pirfenidone on board. And what you can see is a flattening of the slope for most all patients of disease progression in that first year of treatment. And kind of some of their data analyses indicate that the change, the impact of the effect is greater in the fast progressing group than in the slow progressing group. So those are the clinical data. As we visually inspect then the SimPops, you can see the same sort of thing, right? That the magnitude of change in the reduction of slope of disease progression is, the magnitude of change is greater, i.e. the impact of pirfenidone is perhaps greater in the fast progressors than in the slow progressors. Again, we hope validating not only the patients, but the representation of pirfenidone. And then as we transition to Nintedanib, we applied a similar sort of approach. In this case, it's developed by Boehringer Ingelheim, and it's a small molecule tyrosine kinase inhibitor that has, again, a multitude of effects, including inhibiting fibroblast proliferation, as well as procollagen synthesis. There are as well evidence for additional potential effects, but we've thus far only implemented these two particularly impactful, and again, where the data are irrefutable in terms of their contributions. Clinically, these phase 3 data are not unlike what we see with pirfenidone in that it certainly helps slow the rate of progression, but does not arrest it. Another important thing to note here is if you just look at the placebo group, the delta or the change in FVC over that 1 year of no treatment is about minus 200 milliliters, whereas in the pirfenidone study, it was minus 400. So a bit of apples and oranges in terms of the types of patients that were recruited for the study. And that may impact the demonstration of efficacy. So again, we selected SimPops patients that gave us a similar sort of response in the untreated or placebo state, that minus approximately 200 milliliters of FVC over that 1 year. Applied the Nintedanib treatment, and you can see that, again, we're able to generate a nice set of predicted change in FVC, again, slowing the disease progression, not arresting it, and doing so in a way quantitatively that is pretty comparable to the clinical data as well. So with that, let's switch back over to the software and look at our results of this comparison between the three. Remember, one patient that's been treated with Nintedanib, one patient treated pirfenidone, one not treated. And so again, if we go to the forced vital capacity, FVC, what we'll see is for each patient, a difference in response. And so you can see that the untreated patient had a steeper slope than the Nintedanib treated patient that had a less steep slope when treated with pirfenidone. Again, we can look at in this case, let's look at the fibroblastic foci and the rate of progression of that. So fibroblastic foci dynamic fraction. And again, make sure that we have each of the curves described as to which of the states they are. And so consistent with the data that we saw with the respiration, again, it kind of goes stepwise from untreated to nintedanib to pirfenidone. In this particular patient, that helps kind of, it's clear that they're more responsive to pirfenidone, although that doesn't necessarily have to be the case for all patients. So it's a nice, hopefully a nice demonstration of one way that we can compare the various standards of care and treatment modalities versus the untreated. So now returning back to the slides, again, I told you we started with this standards of care, but also to give you a sense of how we might build in a novel pathway, I thought we would then come back to this autotaxin inhibitor. So this is a therapeutic that was actually, I think, was under development for quite a while, but I think it was actually just terminated recently. Nonetheless, it really showed great promise, and it starts with this LPA receptor that then interacts with a variety of pathways, and certainly in myofibroblasts. One key pathway mechanism that LPA really seems to participate in is the proliferation of fibroblasts. So another important aspect here is the substrate LPA is generated via the actions of this enzyme called autotaxin, or ATX. So the treatment approach is, if we inhibit autotaxin, we should have a reduction in LPA, a reduction in the signaling from the LPA receptor, a reduction in fibroblast proliferation. And in fact, the phase 2 data for one ATX inhibitor, this GLPG 1690, really showed over a 12-week study a nice difference relative to the placebo in terms of really slowing and potentially arresting the rate of progression. And so then now the question is, if you recall, the diagrams that I showed you previously, autotaxin, the target, wasn't listed. So how would we represent an autotaxin inhibitor? How could we build in within the flexible framework these pathways? So if our objective is to introduce an autotaxin inhibitor, really what we would start with is building in the LPA receptor and how the ligand then contributes to fibroblast proliferation. And then, of course, similarly, how autotaxin allows for the generation of LPA, and then put the drug on board, slowing down the synthesis or reducing the amount of substrate for the receptor, hence the impact on proliferation, reducing proliferation. And then, you're able to see potentially, by employing these mechanisms, the sorts of phase 2 responses that I showed in the previous slide. So this is the sort of approach that we take, obviously, working with our collaborators or public information to identify quantitatively, what's the Ki of this compound, how much does LPC accumulate when ATX is, characterizing the quantitative aspects of these pathways such that we can introduce it and simulate them as novel compounds. So now let's wrap up by talking about the licensing and services. We, again, in the very near future, will make IPFsym version 1A available to be licensed, with the terms that you see here. I think importantly, it includes 10 hours of training, to help people get up to speed with not only the point and click aspects, but maybe the disease and, a little more in depth how we've, written the equations to describe the pathophysiology that we've just discussed here today. And another really important aspect is that all equations can be viewed and modified by licensees. Again, it's sort of a requirement as we consider implementing novel targets and the collection of in-house data and building out a real platform. Also, we have the ability, DILIsym services within Simulations Plus, to conduct services projects, for us to use IPFsym on your behalf, if you will. So to do that, we kind of really work together and establish a project that will really help provide a positive impact on the program, determine what the resource costs are to execute that program, and of course then the project costs are dependent upon that. it's also worth noting that this isn't one isn't exclusive of the other. In fact, we see frequently, especially as we introduce a QSP model like IPFsym to a new partner, where they will do not only a licensing but also a services project, and then add on an additional component, which is extensive training. So sometimes I describe it as the medical school: learn one, do one, teach one, so that by the conclusion of the project, really, the in-house modeler has a much better sense, a really good sense of how to kind of take the baton and run the next set of simulations in-house. So at this point, I'll wrap it up and avail myself to questions if there are any from the gallery. So.
Thanks, Scott. And as a reminder, if you haven't already done so, you may send your written questions for the group using the questions pane on your control panel. And at this time, I'd like to invite Brett Howell, President of DILIsym Services, as today's Q&A moderator. Welcome, Brett. What do you have for us so far?
Yes, thank you, Arlene. We have some excellent questions that have come in today. We may not get through them all, but we'll give it our best shot. So first question for you, Scott. In the Simulation Plus family, what is the best simulation tool to estimate drug concentration in cardiac tissue?
Oh, that's a great question. I think it's probably GastroPlus. Although, there are a variety of options, but I think GastroPlus is probably the most effective at predicting the concentrations of compounds within tissues, specifically in this case, the cardiac tissue.
Yes, and I would just add to that we also similarly have used GastroPlus and frequently do to predict concentrations in lung tissues, so relevant for the webinar today as well. Second question, how do you evaluate or fit every kinetic constant associated with each of the reactions in the biochemical networks you show in your model, and how can you assure identifiability and uniqueness of the values you find? And I'll stop there, and then there's a second part to the question later.
Sure, it's an excellent question, and in reality, I think it's really an issue that is somewhat unique to these larger QSP models, where there are a lot of parameters, and there are, it can be challenging to have kind of one simple optimization procedure identify all those parameters. So instead of applying something that may not work terribly well, sometimes it does, but not always. Another approach is to kind of do it step by step. And so, for instance, the inflammation component. So we identified a number of studies, a number of data sets that allowed us to characterize, to quantify individual components within that submodel. Same thing with, say, fibroblast activation, with collagen synthesis. And so we're able to then kind of, in a real kind of reductionist way, quantify the various parameters relevant to each of those processes. And then as we pull together those processes and allow them to interact, sometimes there's some mod adjustment to those parameter values. And as those submodels, and like the pathophysiology indicates, interact, there may be some further adjustments. In the end, as you see, I think the real strength of the system is to be able to compare against patient data of all categories to really then help validate that those solutions, that those parametric combinations are generating patients that are, meeting the constraints, i.e., having outputs that are consistent with what you see in the real world. Again, that step-by-step approach is incumbent upon these larger QSP models, I think, for the very challenges that you described in your question.
Thank you, Scott. Does IPFsym use SymBiology or Simulink? For example, does it need, or as a part of that, does it need SymBiology to run, and can it interface with SymBiology if someone needed or wanted it to?
Yeah, at the present time, it is only linked with MATLAB proper, and there is no requirement or, I think, compatibility with SymBiology or Simulink. To be honest, we haven't really explored if there's an easy way to convert into either of those platforms as of yet. Another thing that I'll point out, and it's probably more for kind of version 2, a later version, is we're also exploring potentially migrating it into a more C++ and or Julia based environment. Not for version 1A, maybe for a later version, but I just thought I'd plant that seed here as well.
Thank you, Scott. There are multiple number of antifibrotics in development acting via different pathways. To what extent can this model be used to evaluate other investigational MOAs apart from fibroblast activation or procollagen synthesis?
Well, I mean, an excellent question, and it's it really gets to kind of the crux of the utility of QSP modeling. By encoding all those mechanisms that are relevant for the pathophysiology, it allows a user, a modeler, to explore how a how a compound could have therapeutic potential by modulating those mechanisms. So certainly, if you were to modify macrophage function, apply something of a, let's just call it an anti-inflammatory sort of approach, you would be able to then predict its utility as a therapeutic, its potential efficacy for treating IPF patients. So for all the mechanisms listed in or described within IPFsym, you have the ability to explore each of those targets as potential means for treatment.
Great, thank you. This person says, very nice work. Can you please comment on how you bridged from the biology represented to outcomes like FVC? Does your virtual population include variability in parameters in this relationship specifically?
That's another great question. In this case, for the respiration, we really relied on kind of the, so I described earlier that the underlying pathophysiology, the biochemical changes, can manifest as changes to the anatomy, i.e. that honeycomb tissue. And that honeycomb tissue is comparable, it's kind of like dead, non-functional tissue, a la a lung resection. And so by looking into the lung resection literature, we were able to kind of establish the relationship between kind of the amount of normal parenchymal tissue and FVC for a person of a given size. And so we really were able to rely on those sorts of data sets to link then, to the extent that a simulated patient has N fibroblastic foci volume and X fibroblastic foci volume and Y honeycomb volume, we're able to then predict, again, relying on kind of those anatomical sort of base data, what the respiratory impact would be. So yes, lots of variability in the change in each of those volumes, but really then tethering it back to those data sets describing anatomy back to the physiologic outputs.
Great, thank you. So this will be our last question. How are the treatment effects of nintedanib and pirfenidone built into the model, i.e. as symptomatic or disease-modifying effects?
So they are disease-modifying effects. And I neglected to mention that we also have PBPK models of each nintedanib and pirfenidone predicting the concentrations within the lungs. And then they go in and, once they're at the site of the target, modifying the function of the fibroblasts, be it collagen synthesis, activation, or proliferation. And then as those are reduced in a given IPF patient, then obviously the disease progression slows and there's an amelioration to some extent of the extent of the disease.
Great, well, thank you very much.
Well, thank you, Brett and Scott, and thank you everyone for joining us. I hope that you've learned how using predictions with QSP modeling software like IPFsym will make the clinical development process more efficient for your organization. We invite you to learn more about IPFsym on our website at www.simulations-plus.com. And you can also find Scott and Brett as presenters at the 2021 MIDD Plus Scientific Conference this coming March 3rd and 4th. This concludes our webinar for today. It has been recorded for playback and will be available on our website and YouTube channel. Have a great day, everyone.